As you heard, I'm Ellen Gravelisi. I'm joined by Alexandria Hammond and our panelists, and I'm going to ask our panelists to introduce themselves and their companies. So we'll start with Deb Garrity.
Hi, I'm Deb Garrity. I'm the CEO of Enochion. We are a phase 2 clinical stage company developing novel therapeutics for individuals with autoimmune disease. We harness natural pathways in the liver to drive what is known as immune tolerance, or the state of unresponsiveness by the immune system. Thank you for having me.
Thank you. And Sam?
Great. Thank you so much for having me here as well. I'm Samantha Singer. I'm the president and CEO at Abata Therapeutics. What we're doing is we're creating transformative therapies for patients who have serious autoimmune disease. And we're doing that by creating a new class of regulatory T-cell therapies that are derived from a patient's own cells. We have a distinctive strategy that uses natural Tregs engineered with a T-cell receptor, so we have tissue-targeted activation. Our lead program is progressive MS. We expect to be in the clinic next year, and we're fast following behind that with type 1 diabetes. And what we envision is that an autologous Treg cell therapy could deliver durable curative benefit to patients in a single dose because it's available to expand and regulate the immune system for a very long time, possibly even a lifetime.
Thank you so much. So I want to thank Steve Greenberg for giving that beautiful introduction to what's happened in the immunology space from the advent of corticosteroids all the way up to monoclonal antibody therapies. And in our field, in the immune-mediated and autoimmune diseases, we've had amazing advances, as you probably know. Most of those advances have been with the use of monoclonal antibodies that are directed against pro-inflammatory cytokines. And although they've been transformative, what they do is they interrupt the effector loops, and if stopped, the disease typically recurs. And so, although disease remission is really the goal of therapy in our patients, it's rarely achieved, and we don't have any cures. So the question that we're asking today in this panel is, can we reprogram the immune system to restore immune tolerance? And we've taken a page from the playbook of the oncologists, using both cellular therapies and trying to manipulate immune pathways to treat our diseases. And so today we're going to be exploring priorities, opportunities, and possible barriers to the success of these approaches. And we're going to cover three major areas. The first is approaches to T-cell modification, as we heard from Sam Singer. We're going to talk a little bit about CAR T-cells and then mostly about T regulatory cells. We're going to think about how we might harness the immune cells from healthy donors and give them to our patients, and then manipulation of immune pathways, for example, the checkpoint pathway. And then finally, we really want to think about the population of patients that we're treating. This is a very different population from the cancer patients. These are patients who have disease that is life-threatening, but not immediately life-threatening. And so, it brings in questions about trial design and kind of the bar for safety in our patients. And so I'd like to begin by asking finally our final panelist to introduce herself.
Hi there. Sorry for being a little bit late. Do you want me to get started?
Just your name and your company, a little bit about what your company does.
Great. So my name's Diana Brainerd, and I'm the CEO of Alavir out in Waltham, which partially explains the delay this morning. What we do is off-the-shelf allogeneic virus-specific T cells. So it's gene-edited cell therapy for viruses that affect mostly immunocompromised patients. So harnessing immune cells to replace the deficit that's causing patients to suffer from these diseases. And we're in the clinic right now and look forward to sharing more.
Okay, thank you very much. So we're going to start with approaches to T-cell modification. And recently in our field, there have been some very preliminary studies using CAR T-cells. There's a very successful study using a CD19 CAR T-cell in refractory systemic lupus erythematosus. And the interesting thing is that this was very effective even though this patient had taken rituximab and had failed with rituximab. So it's also been treated in a very minimal way, I would say a few patients, with myositis and systemic sclerosis. And so far, the CAR Ts have a very good safety profile in our diseases, and that first patient that was treated has been in remission for up to 2 years, drug-free. So I'm going to turn first to Samantha Singer to tell us a little bit about CAR T cells, the modifications that need to be used for CAR T, how have they been applied to immune-mediated diseases, and tell us about other approaches such as Tregs.
Sure, absolutely. So I'd open by saying that we're living in a very exciting time because we have the ability now to harness cells for their natural abilities, right? And cells such as Tregs have a polypharmacy to choose from, and they can be very, very responsive to whatever specific tissue that they're in. It goes far beyond what you could try to do with individual agents in some kind of combination, and so you can kind of pack multiple drugs into one tissue-specific therapy that again can be really responsive to the needs of the tissue that it's in. So it's a very exciting time to be able to do that. When you're comparing different types of approaches, one way to think about it is through what the product design is. And I would break it into 3 different parts. There's the chassis, there's the targeting moiety, and then there's what I'd call add-ons. So for the chassis, that's the choice of what cell type you use, and that basically dictates the fundamental nature of your therapy. And of course, different cell types are going to have different types of outcomes and risks. So in oncology, everyone's very familiar with the CAR Ts. These are T effector cells, and they are phenotypically programmed to destroy other cells. They've also been used effectively in lupus as well. So if there's a subset of cell types that you want to get rid of, that's a cell type, a chassis that you might choose to use. Regulatory T cells, on the other hand, they are phenotypically programmed to quell inflammation, to restore tissue homeostasis, and to promote repair. Pretty much the polar opposite of an effector T cell. And so, there also you get different kinds of risk profiles. So when you look at T effectors, a lot of times people think of cytokine release syndrome, right? But a regulatory T cell, on the other hand, is incapable of creating that kind of cytokine storm. So it's not necessarily a risk that you need to watch for. So the chassis itself, again, both gives you different outcomes and different risks. The targeting then dictates where and how your cell gets activated. And there are different pros and cons for a CAR versus a TCR. And so one aspect to think about is the signaling pathway. So a T cell receptor uses the native signaling pathway, and so it operates at physiological amplitude and intensity and regulation. Makes it a little bit more predictable, a bit more responsive to the surrounding environment. A CAR piggybacks onto a signaling pathway. There's some risk there then for non-physiological activity or perhaps even gaps in repertoire. Now the trade-off is that TCR is a naturally occurring protein, so you have to discover it, like a needle in a haystack approach. And so we did have to invest a lot to build that capability at Abada. In contrast with a CAR, you can engineer it, and that has advantages for time and risk and investment. Another way to think about it is how does each of them see their target peptide? Right? So a TCR sees its target antigen by docking with an antigen-presenting cell. That APC-TCR interaction is a big part of what enables a Treg to be a Treg. Tregs, one of the many things they do is they downregulate antigen presentation on those APCs, and that can also lead them to become tissue resident. So a CAR recognizes a peptide on another cell surface, so it kind of misses that APC interaction piece. Again, the trade-offs there, though, are a TCR, you have to match it to a genetic HLA haplotype so you can get proper docking. And so at Abada, we've chosen indications that have a genetic link with a specific HLA. CAR can actually be used for any patient and is not limited by that kind of genetic background. The last kind of compare-contrast I'll make is that a CAR tends to have higher affinity, while a TCR has higher sensitivity generally, so it can be more responsive to a lower density of antigen. And so you can kind of see how, depending on the biology you're targeting, the disease, what your objectives are, why you might choose one or the other. And I think that's what kind of really drives it. And then the last piece when you're looking at different products is the add-ons. right? So you might want to engineer into your therapy some kind of capability that it doesn't naturally have. For example, if you have a strong safety risk, you might want to put in a kill switch so you can turn it off whenever you'd like. I would say that you want to make sure when you're thinking about add-ons that you look at the biology and that you know exactly what problem you're trying to solve, because anything that you add on does create technological, biological, and safety risk. So I think it's not something you want to do without having sort of specific rationale. At Ibotta, we actually don't engineer anything other than the TCR in. We think that Tregs, when you look at the biology, are just exquisitely well suited for the job that we want them to do. And so you also mentioned using the tools and technologies that are already developed for CAR-Ts. They're incredibly enabling. We're essentially using all established technology so we can move quickly to the clinic. We've partnered with Elevate Bio, which I know the AlloVir team is also very familiar with. They have a lot of that experience. It helps us move quickly. It means that we can benefit from all the advances that are happening in other companies that are doing autologous cell therapies. Some of you might have seen that the Legend CEO a couple weeks ago said they're going to be capable of delivering thousands of doses to patients by the end of 2025. That's remarkable, and so we can draft on that kind of improvement. It's also helpful from a corporate strategy and planning perspective, because you can use what you know already about timelines and risks and investments to plan ahead. And for small companies in particular, knowing what your sort of cost and runway and risk is incredibly important. So I think we'll really benefit quite a bit from what's already in place.
Great, thank you. So you mentioned a study in lupus, and I think you're referring to the study with a patient with skin disease, severe skin disease in lupus, and that was autologous expanded T cells that migrated to the skin and attenuated interferon gamma production. I wonder if you can talk a little bit about how scalable are the use of autologous Tregs?
Yeah, absolutely. So with today's technology, it's pretty ambitious to think about scaling to thousands and thousands of patients, but there's a tremendous amount of opportunity in that. So there's a lot of process improvements, a lot of economies of scale that can be built in. We've seen across new modalities of all kinds this happening across protein therapeutics, RNA, and even the cell therapies that are in the clinic now are incredibly more scalable than what they were before. So we're investing to advance that. That's part of why we select Elevate Bio as well, because they have experience in that. And you do see the companies with autologous cell therapies that are in commercial asserting that they think within a matter of years that they can get to that scale. So I think it is highly scalable with investment in the direction we want to go.
And can you tell us a little bit about the challenges in preparing these cells? For example, the purity of the cells, how do you measure the purity, and also the challenges with expansion methods?
Yeah, sure. So purity is incredibly important for what we're trying to do. So in oncology, when you just need just enough effector T cells to go and kill the tumor, it's not as important. It's important to a certain level, but not to the same degree. For a regulatory T cell therapy, what you don't want is you really don't want many, if any, T effector cells in there, especially when we're adding in a TCR that sends it to be activated right at the site of disease. So if a effector T cell gets that TCR, we're actually causing disease instead of ameliorating it. So purity is incredibly important, and that's where we invested a tremendous amount of effort and innovation with Elevate Bio to create a process where we can purify thymically matured, terminally differentiated, phenotypically stable Tregs to very, very high purity. In fact, we are regularly achieving over 95% purity on our Tregs as measured by a gold standard epigenetic marker. So it is incredibly, incredibly important and also very, very difficult to do. And so that's where we've invested a lot of our innovation.
And just one final question. I know that in the early days of infusing Tregs, some of those Tregs could actually morph into effector cells. Has that been taken care of, that problem?
Yeah, so there's definitely data out there that suggests that you can get phenotypically unstable Tregs. Those are a certain type of Tregs where that's part of their arsenals, that they can flip back and forth. These are peripherally derived Tregs. They are phenotypically different in that way. Their expression profiles are different. Their epigenetic markers are different. Their cell surface markers, are different. So the Tregs that you really want are thymically matured. They can be differentiated by certain cell markers. Numerous groups, including our company founder Diane Mathis, but many others, have shown that this particular type of Treg is incredibly phenotypically stable, even under highly inflammatory conditions. And in fact, in our own lab, we've pressure tested the product of our manufacturing process under inflammatory conditions in vitro for well over a week, and these cells remain stable. So if you get a certain type of Treg, yes, it can phenotypically switch. If you get the right kind of Treg, it does not, and it is locked in with that phenotype.
Okay, thank you. It's incredibly exciting.
It is.
We're really excited about this for our field. Next, I'm going to turn to Deb Garrity. Your company uses a different approach to really restore normal immune tolerance, and you're targeting autoreactive cells for removal and using therapeutically engineered proteins and peptides. So I wonder if you can tell us a little bit about that, and specifically the use in celiac disease, diabetes, and multiple sclerosis.
Yeah, thank you. It's actually a pleasure to be on the panel with a bunch of other women, actually, who are running companies. I think it's a really exciting time. You mentioned this, Samantha, as well in your talk for immunology. I liken a lot to, when I go out and talk about the company, to probably about 5-10 years ago about IO. There was a lot of biological progress. There was a lot of insights that got, turned into drug development. And I think we're at that time, at that precipice for autoimmune disease, and really thinking about ways to address the immune system in unique and different ways. Enochion, if you think about it, immune tolerance is something we live with every day. We have red blood cells, they are recycled, and they are broken down, and your body doesn't recognize them as foreign, they're recognized as self. That is unresponsiveness to what could be termed as a foreign antigen or foreign protein, et cetera. And so we basically discovered what we thought and what we believe is a novel approach to driving immune tolerance, particularly in our lead program, which is in phase 2 clinical development in celiac disease. We have a program in MS where we just finished enrollment in multiple sclerosis, and then we also have a program kind of behind that, ready to enter the clinic for type 1 diabetes. Well, what is immune tolerance? I think it speaks to a lot of the things you just spoke about. We, by harnessing this pathway, which is through a specific receptor that sits in the liver, that's how your body breaks down red blood cells. It gets taken up by the liver, it gets presented appropriately, and then the body says, all right, don't attack your red blood cells. And so that's marked by 3 really important attributes. That's deleting those T cells, those nefarious T cells that would attack either the tissue or the organ. So let's say for type 1 diabetes, it's the pancreas. For celiac disease, it's the gut. We also, those T cells that might remain that we don't get to put through apoptosis, they're energetic. So we upregulate markers of energy, including, as you talk about, checkpoint, upregulation of PD-1, LAG3. These are important markers that are leveraged and targeted for oncology. And then we also upregulate T regulatory cells. We do this naturally, though. And these are, as Samantha said, really important and actually really sought after ways to drive long-term memory, long-term durability, and therapeutic benefit peripherally as well. So we upregulate these T cells. Even though we're hitting the liver, we're having very broad, very interesting regulatory effects in the body. So for example, in celiac disease, we presented data last year at DDW showing that our platform, when we deliver a particular immunodominant domain of gluten, which is called gliadin, we could basically knock down nefarious T cells, and delete those. And we know this because we went back in and we looked at their phenotype, and they have markers of upregulated PD-1 and LAG3 on the cell surface. So that was very exciting to us. We also generated data recently with MS, where it's clear we're driving a concept which is related to the T regulatory component of our mechanism of bystander suppression. What does that mean? A lot of these autoimmune disorders have more than one antigen involved. So MS, I think the latest date was there's maybe 7, maybe 9, maybe 11. But in our lead program, we have one antigen, which is a component of the myelin, and we've been able to demonstrate a reduction in T cell reactivity to related antigens that are not necessarily in the drug itself. So I think for us, this is really exciting for the celiac disease community, as well as for the MS community, type 1 community as well. Celiac, there's nothing currently in development. Think about a celiac patient. I know everyone says, oh, gluten-free diet, they can just avoid gluten. It just doesn't work that way. I mean, I'm sure many of you either have family, children, friends that have celiac disease. You go to a hotel, you go, right, you're in the West End, you're staying and go have the breakfast. Well, they put flour in eggs, and they may not know that. It makes them look pretty and fluffy. So you eat gluten that's in an egg, which you would never think, and you end up having a pretty severe, GI issue. So I think for those patients, it's really exciting. For MS patients, what's exciting for us is, it's a heavy lift. I think MS has had a lot of therapeutics developed for it, but finding something that could provide long-term durable benefits, where you're really retraining the body to say, hey, wait, take a step back, don't go after your myelin sheath. Now you have to get in at a certain time, and I think for you, the same with diabetes, and I think the Prevention Bio study shows that early intervention is very, very important, and so for us, you want to get in early enough so you don't have complete destruction. I think additional, from a celiac perspective, we're seeing in the space some very interesting things with other immune tolerance players, where they show a reduction in the damage to the gut. That's another marker sort of efficacy, and you wouldn't think that a celiac patient has issues, but in the long run, they have a much harder time absorbing nutrients, getting the kinds of vitamins and things like that from the foodstuffs. So to be able to protect the gut, which is one of the endpoints that we're going to be measuring in one of our 2 phase 2 studies, is a really key component of that. And then finally, really thinking about symptoms, right? Can you imagine being on a plane, getting an accidental exposure, and then, you get an accidental, taste of something that's got gluten in it, right? Ketchup has gluten in it, and you're sitting there on a plane. So to be able to provide something that really harnesses the body's own system without the need for, CAR T, without the need for delivering something else that upregulates that T regulatory component. One of the things we've been able to do is take the data from those patients, take the data from the studies that we've done, and we've integrated that into how we've continued to learn and build new IP. And we've developed antibodies, for example, that target the same receptor, but we believe that they can work without having the specific antigen that's important for the disease. It opens up a lot of possibilities for us, such as IBD, Crohn's, et cetera. And it conducts, the way we think about this, it conducts the immune system to go to that peripheral location, either the target organ, the target tissue, et cetera. So it really offers, the way that we're thinking about this, it offers a safe approach, a tolerated approach. We have yet to actually hit a maximum tolerated dose in any of our studies, which is very exciting for patients, because a lot of these products, right, that have global suppression of the immune system can be problematic. I think they mentioned it in one of the earlier panels, that they're at risk for other infections and things like that. So it's pretty exciting. I think it's a great time for this field and for those individuals who have these disorders. I think it's early days, but we continue to learn, and I think the biology, really understanding the mechanisms that drive this, much like with IO, we're at the very earliest of stages, but I think there's a huge amount of value for investors. It's a huge amount of value for those companies who are doing it, but there's enormous value for patients and families in the long run.
Yeah, thanks so much. Really exciting work.
Very exciting.
I'm going to turn things over now to Alex to talk with Diana Brainerd.
Thanks, Ellen, and thanks everybody for joining us. This is a great backdrop for our next topic, which is harnessing the immune system to treat viral infections. As been discussed today, there's been a rise of patients that are immunocompromised or immunosuppressed, or have periods of vulnerability from, let's say, a conditioning regimen. So with that, patients are very susceptible to viral infections. There's been a lot of great innovation of late, especially with virus-specific T cells, which have a off-the-shelf nature and can potentially target multiple viruses. So with that, Diana, can you give us a brief overview of virus-specific T cells and your platform?
Yeah, absolutely. That was a great introduction. Thank you. So, I think that the recognizing that immunology is really at the heart of so many different diseases, right? And sometimes it's very obvious, right? Autoimmune disease. I think there's a recognition now that maybe wasn't there a few decades ago around oncology and having a huge immunology component. And infectious diseases is one area where it also makes perfect sense. There are many diseases or viruses or microbes that we're exposed to that don't pose threats and in fact can be even beneficial in some senses. But for immunocompromised patients, when you take away part of their immune system, you create a vulnerability that can cause a lot of problems. And the concept of replacing the immune deficit that's causing this problem has been around for a long time. About 20 plus years ago, the first publication using an autologous approach to give somebody T cells who was having post-stem cell transplant of progressive viral disease to sort of control that disease was published in Science. And since then, there have been a couple of really critical advances in the concept of using virus-specific T cells to control viral infections. The first is the recognition that you don't need full HLA matching. So when you're targeting virus-infected cells, you really only need to match at two or more HLA loci to have efficacy. And you don't lose on safety because these cells are exquisitely specific for viral antigens. And so that enables you to have an off-the-shelf product, which means you can get those cells to patients faster than if you have to create an autologous product, which then can be too late for a patient with a rapidly progressive viral disease. The other really important innovation was this recognition that in addition to a T cell product that's targeting one virus, under the right culture conditions, you can create a product that targets multiple viruses. So any given cell will be specific for one virus, but you can grow and expand the cells in the right conditions with the right peptides, the right cytokines to come out on the back end with a mix of T cells that are targeting different viruses. And at AlloVir, this is what we've done with our lead product targeting the 6 most commonly impacting viruses that cause disease in the transplant arena, so either stem cell transplant or solid organ transplant. And in this way, you can go for treatment of any one of these 6 viral diseases, but very importantly, you can also pivot to a preventative approach, which for particularly certain types of stem cell transplant patients, allo stem cell transplant patients who have a very high risk of reactivating one or more of these infections in their 100 to 180 day window following transplant before their new immune system has engrafted. If you can keep them covered with our T cells that are protecting them, then that allows them to develop their own immune system and really move along on their pathway to recovery. And so, having an option like this for treatment and importantly for prevention is important, and it's exciting, and it makes sense too in the conceptually when you think about what we're doing is we're giving the patient the cells that they're unable to create on their own and covering them for that window until they can make those cells. And the toxicity there, as opposed to some of the currently available antivirals, is much lower because you're not impacting renal function, you're not impacting liver function, you're not delaying engraftment or lowering cell counts in this really critical time period. So, it's an incredibly simple concept, but the turning something from a concept and an academic exercise into a product, I think has really been enabled by the advances in cell therapy overall, the manufacturing, some of the scaling that's been done across the field, we've really been able to leverage that so that instead of having a product that takes weeks and weeks to make, you can create an off-the-shelf product, you can store it, and then more and more there are more capabilities for distribution, for storage. All of these things are really important when you think about how to create access beyond just one academic center, but really something that could be globally available and used on a regular basis for patients.
Thank you. And what about the durability of these type of products? And what about the risk of allo rejection?
Yeah, so I think with virus-specific T cells, the goal is to cover the patient when they're either at risk for the infection, if you're taking a preventative approach, or for the period that they're having their infection until they have recovery. We're not trying to permanently engraft these cells. And so the durability is driven by two factors. One, the presence of viral antigen, what causes these cells when they're given to expand. It's a living product. So the cells that are specific for the virus will expand in the presence of viral antigen, and that will drive survival. And then the other key piece is the status of the host immune response. And so when there's immunocompromise, our cells can persist for longer in spite of that HLA mismatch. If a patient has a normal immune system and doesn't have viral antigen circulating, we would not expect our cells to last beyond 2 to 4 weeks, but that's okay because our cells wouldn't be needed in that scenario. On the other side of survivability is the question of reactivity, alloreactivity, and so our cells in vitro have not demonstrated any alloreactivity because they are so, at the end of this culture period, so exquisitely viral specific, CD4 and CD8, with very minimal contributions of other cell types. And what has been seen in the field and what we've seen in our clinical data is really no signal suggesting there's graft versus host disease, or in the solid organ transplant space, evidence that there's any sign of our cells attacking the organ. And again, this is because of the exquisite specificity of our cells. And in fact, there's an interesting relationship in graft versus host disease between infection and graft versus host disease, where they sort of amplify each other. Each is a risk factor for the other. And our phase 2 data, which is uncontrolled but demonstrated lower rates numerically of graft versus host disease than you might have expected in this population, and raising the concept that if you can, again, take infection out of the equation, maybe you can sort of reset that immune environment and lower the risk of graft versus host disease as well as other complications. So that remains to be played out in our phase 3 placebo-controlled trials that we have ongoing. But again, it is an exciting concept, again, this idea of reprogramming the immune system. You can solve for the problem right in front of you, but may also have some other ancillary benefits as well.
Thank you so much. So in the remaining few minutes, I'd like to talk a little bit about inhibition or modification of immune pathways. For example, obviously in the cancer field, there's been great success with inhibiting the checkpoint pathways. And there's been a recent publication in our field doing the opposite, to actually augment T-cell checkpoints. That was a PD-1 agonist that was used successfully in a phase 2 in rheumatoid arthritis. And so I'm wondering if you could tell us a little bit, and I'll open this to all of the panelists, about ways in which we might modulate immune pathways that are present for the benefit of patients with autoimmune diseases.
Sure. How about I'll start. I alluded to this earlier. I think, and it kind of makes sense, right? You're going to inhibit something in one setting, and the thinking is, well, let's turn that on its head and do it in a different way. And I think as we've continued to really interrogate the pathway, we've taken the patient samples. Our phase 1 was done in patients. Our phase 2, ongoing phase 2 studies are in patients. We take either biopsies, we take tissues, we take blood, PBMCs, et cetera. And what we've been able to show is, and I mentioned this earlier, and we presented some very exciting data at DDW earlier this year, where we drove upregulation of PD-1, which has these really, I think, interesting benefits. I think you have to think about it carefully, right? Because you don't want to just sort of unleash, upregulated PD-1 everywhere. And so for us, we do this by using the pathway to do it in a very specific way, either through an antigen that's specific to that disease. So for us, it's gliadin. I think, but we also do it by targeting a specific tissue so that you're really honing in on, so for example, IBD, it would be the gut. And it makes a lot of sense. And for us, we're really trying to use the body itself, rather than delivering something that's kind of a foreign, yes, we have a honing mechanism. You talked a little bit about that, Samantha. That's what we're calling a glycosylation signature that targets this receptor. But then that falls apart, that gets broken down, it's gone within the body within about, 3 to 6 hours. And we let the immune system kind of do its job. So to be able to turn that on its head, and I think also build on years of experience of how you can target checkpoints, and there's a safety database that exists with that, just given the years of using PD-1s, I think we'll continue to grow that out. I don't know if you want to add to that, Samantha and Dana.
Yeah, I would say the way we're going about it is rather than going after one specific pathway by using an entire cell, is we're sort of more broadly using multiple pathways. And because the cell can be regulated in its native way, Tregs are really well known as being sort of tissue resident. That's their happy place. They move around the body till they find an antigen that it recognizes, moves into that tissue, and then is responsive to whatever that tissue needs. And so whatever pathway needs to be activated for that tissue to be healthy, to have immune tolerance, to be repaired, gets activated and chosen essentially by that Treg in place.
Okay, thank you. So we have only a few minutes left, but I'd be very interested to hear your thoughts about the patient population that we're treating. Again, these are patients who have life-threatening diseases, but not immediately life-threatening. And we're now going to bring in these new cell types that are new, novel, or we're going to be manipulating their immune pathways. How do you think about safety in these patients, and also trial design? Does it affect trial design that these patients are not imminently dying?
Yeah, I might start with that, but then trial design should probably go to the folks that right now are in the clinic. It'd be great to hear from them. It is important because you have to have a higher benefit-risk ratio for patients that have these long diseases. They are devastating, and make no doubt, these diseases do kill people. It's just over a very long time. So in type 1 diabetes, if you're diagnosed as a kid, you lose a decade at least of your lifespan. So have no doubt, they are deadly, but the fact is they do live with them for a very long time. And so having a therapy that has debilitating side effects or a very high risk of sudden death is just not going to be feasible or acceptable. And you also have to really think hard about what the therapy is going to do over very long-term exposure. So, if treating type 1 diabetics, they're going to be exposed to this for literally decades, right? And so you do have to have a much higher safety bar. It affects who you choose as your target population, especially while it's still unproven. So Tregs are being tried as a cell therapy in patients for whom everything's failed or they have no options. You also have to think about in product design. That's why a lot of my comments have been really directed towards how do you optimize the safety of the product and thinking very hard about that, so your product design choices are featured there. And then there are clinical trial considerations and sort of how important it is to do biomarkers to watch for damage, as well as to make sure you're characterizing your product that it's doing what it should. How are you thinking about it?
Yeah, I mean, I think I'll go quickly because I want to give Diana a minute. I think for us, obviously, because we target a receptor in the liver, it's really important for us to understand that the liver is protected. And to date, we've had no issues. We have no changes in liver function tests and things like that. And I think in MS in particular, what was very important for us, the history of immune tolerance has shown that you could exacerbate the disease. So for us, it was very, very important to go as high as we could, and we still have not reached an MTD on that, and not to exacerbate disease. And we haven't seen any evidence of that in our study. So those are really key aspects of how we've thought about the study design. Diana? You got a minute.
Thank you. But I can do this. So I think that there's oftentimes the patients are most excited to try something, right?
Yeah.
And it's about demonstrating safety and reassuring the physicians and clinicians that this new technology or new approach is safe for patients and something that giving them comfort with using something novel. Oftentimes, this is seen in cancer as well, but you start with the patients that have kind of exhausted all options. So it might be, for example, our first phase 2 was in patients with refractory and resistant disease. They had nothing left, right? So you show safety and potential signal of efficacy there, and then the patients who can really benefit most are using a prevention approach. They haven't even gotten sick yet, but you're not jumping there first. And you mentioned that earlier with the lupus patient who had failed rituximab, right? Yeah. You go into the scenarios where the benefit risk is maybe a little bit different, even though that's not the ideal usage, right? Ideally, you move further upstream, but you have to take it in a stepwise fashion to get everybody comfortable with the safety, prioritizing that first.
Yeah, thank you so much. Well, unfortunately, we're out of time. It was a fantastic discussion. I want to thank all of the panelists for all of the work that you're doing for our field. We really appreciate it. Thank you.
Thank you.